Okay, good afternoon, everybody. And we'll continue our discussion about bacterial form and function. And this is where we ended up our last classes with a discussion about the structure and function of the cell wall. The cell wall being completely distinct from the cell membrane as a structure. The cell wall is a non-living, very strong entity composed of the molecule peptidoglycan. Peptidoglycan, basically, it's a disaccharide. It has two kinds of sugar units, and they form these long filaments, which forms the backbone, really, of the cell wall. Now, those filaments here are linked to one another by peptides, which link across, by protein units, which link across, forming very strong peptide bonds here, holding these filaments in a rigid and extremely strong sheet. So that is the basic structure of a single layer of the peptidoglycan. But there are elaborations on this in the two great groups of bacteria that we recognize, called the gram-positive bacteria and the gram-negative bacteria. These are two groups which are preceded along entirely different evolutionary lines. And just 1 second, sorry. And if we have a look first at the gram-positive cell wall, here you can see here's a single sheet of the peptidoglycan filaments, all joined by peptides together. But now in the gram-positive, we layer many of those layers on top of one another, one after the other, and tie them together with this particular molecule here called teichoic acid. And teichoic acid ties these all together into a very strong, rigid, net-like structure, and also anchors them to the cell membrane. So the cell membrane and the cell wall are connected, but the cell wall itself is entirely separate, and it is a non-living entity compared to the cell membrane, which you see here below with its embedded proteins. The cell wall is not going to stop the passage or regulate the passage of any substances. It's a very, very porous, open structure. But this is the gram-positive cell wall, very thick. There's only one membrane that we see here, the cell membrane, and the presence of teichoic acid is another diagnostic feature for the gram-positives. And gram-positives include many, many bacteria that you're going to hear about. Many of the rods, Bacillus, for example, is a gram-positive, and almost all of the cocci. It is very rare to have a gram-negative coccus, although they do occur, as I have told you. Most of the cocci are gram-positive. So then the next group to discuss are the gram-negative bacteria. And gram-negatives are extremely interesting and considered to be a more advanced in evolutionary terms than the gram-positives. And they have a completely different kind of structure. So we'll start off with what's similar to the gram-positives. Here's the cell membrane here. Just the normal cell membrane, exactly the same as it occurs in all bacteria and all other cells. A phospholipid bilayer with embedded proteins. Embedded and associated proteins, I should say. In the gram-negatives, there is a very thin layer of peptidoglycan. It's composed, this single layer is composed the same as the single layers of the gram-positives, but we don't have multiple layers, and because we don't have multiple layers, they don't have to be tied together by teichoic acid. Teichoic acid does not occur in the gram-negatives. But what is really strange about the gram-negatives is that they have another membrane. And it's a distinct membrane structure. It looks rather similar to the cell membrane. It is called the outer membrane. And it has the same kind of molecules with lipid fatty acid tails. and water-soluble heads. But these lipids are rather strange in that they have associated with them extensive sugar groups. And we call these, the composition of many of these lipids are lipopolysaccharides. Not all of them, not all of the components here are lipopolysaccharides, but many of them are, and they are referred to as lipid A. And lipid A is here in the outer leaf of this outer membrane. So these, the structure is rather similar to the cell membrane, but in fact, its components are quite distinct. And these components of the outer membrane is lipid A is of enormous clinical significance because the immune system of mammals in particular responds very, very vigorously to the presence of circulating lipid A. It will also respond to a certain extent when it is in the living cell, is bound to the living cell outer membrane to a certain extent, but the really powerful response happens when these components are solubilized. When they begin to go into circulation in the body, they cause such a profound immune response that it leads to the collapse often of the physiological systems of the body. In particular, it leads to a catastrophic drop in blood pressure and often shutting off of blood circulation to the organs and to the extremities. This sounds very strange. Why is the body killing itself in response to the presence of these bacterial components? It is simply an overreaction. It is an overreaction to the presence of a very powerful antigen in circulation and a very dangerous clinical situation indeed. So something I would point out to you, if a clinician is dealing with a gram-negative infection, a gram-negative septicemia, let's say, that is circulating in a person's body, then there is a very difficult clinical choice to make, because our natural response is to say, okay, let's treat with antibiotics and kill off the bacteria. But the problem with doing that is when you kill off the bacteria, they lyse, they burst, and they release these, especially lipid A components, into the circulation, and that occasions the onset of sepsis. And so treatment of a gram-negative infection is completely different to treatment of gram-positives. Gram-positives do not have these strongly antigenic components the way that the gram-negatives do. Gram-negatives have to be, a person with a gram-negative infection has to be treated very judiciously with supportive care and slow administration of antibiotics so that you don't get a catastrophic killing off of the population of bacteria. Okay, so here's the cell membrane. This is the outer membrane. Between them, there is this thin peptidoglycan layer. This here is called the periplasm. Also, you may see it referred to as the periplasmic space. Now, that is almost gel-like composition because it is so full of molecules that have been acquired from the outside. And this is one of the things about gram-negatives. They are very, very good at acquiring molecules, rare molecules, and accumulating them here in the periplasm. And that is the responsibility of integral proteins, which you can see here. And so there are also some peripheral proteins as well associated, but it's the integral proteins which we're particularly interested in, because many of them are transport proteins or porins, and they will specifically transfer specific molecules to the inside. Because gram-negatives are good at accumulating rare nutrients and storing them like this, Gram-negative bacteria are very good growing in highly dilute situations, such as fresh water, seawater, those kind of habitats. When you go there and sample, almost all of the bacteria that you find are gram-negative, because this is an adaptation to low nutrient levels. There are many, many, many gram-negative bacteria that we will encounter. They don't only occur in low nutrient environments, they occur in some very rich nutrient environments. For instance, probably most of our gut microflora are gram-negative. We do have gram-positives as well, but many, many, many of them are gram-negative. And the most famous gram-negative, of course, is Escherichia coli, or E. coli, and its relatives, which are all gut bacteria. Okay, so this is, here is the thing that we're particularly interested in. And this is lipid A, and the lipid A, it has this kind of structure with a soluble head and a hydrophobic tail. And it is referred to as being an endotoxin. An endotoxin is one which is a component of the bacterium itself and tends not to act as a toxin until it is released when the bacterium dies. This is as opposed to an exotoxin. An exotoxin is one which is released by the bacterial cell, by the living bacterial cell, into solution around it. So you can see here that this lipid A has this very long polysaccharide tail. And this O-specific polysaccharide itself is also antigenic. This lipid A that is particularly dangerous. Okay, so we've got those two great groups. It also introduces to you the idea that in the clinical laboratory, very often the very first thing you do is to do a gram stain to determine whether you're dealing with a gram-positive bacterium or with a gram-negative bacterium. It is a very simple staining technique named for the man who developed it, who was Graham. And it depends largely upon the structure of the cell wall, on the thickness, and one or two other things that I'll mention in a minute to distinguish between the two groups. And this is the procedure. You will do this in lab if you haven't done it already. And it's a simple stain technique. The first thing we do is to stain. First of all, you have to make a smear. So what you want to do, you make a suspension of bacteria. You can take from an agar plate, or you can use a broth, but you always have to be very careful that it is dilute, that you have it so dilute and you spread your drops so wide that bacteria are well separated from one another. If you do it too thick, they lie on top of one another like this. And all of the capsule proteins and everything else that we know about, they all die, and they make it very difficult to interpret the results. So it's easy, but it has to be done precisely. So the first thing we do, we dry that. We actually flame dry it, or heat dry it rather. And when you that is called heat fixation. What happens is you actually bake the bacteria onto the slide, you kill them, but you also fix them. Their proteins adhere to the surface, to the glass, and stick there, so they don't wash off. You apply a dye called crystal violet. It's deep purple color, and crystal violet is just a general stain. We can use it just in routine if we want to stain cells to see their shape, etc., etc. But here it's being used for a special purpose. So we initially stain with crystal violet, and we then apply an iodine solution. And this is a mordant. What that means is it ties the crystal violet molecules tightly to the bacterial cell, and especially to the components of the cell wall. Also stains the interior, it stains the cytoplasm and everything as well. Now what we do is, this is the critical step in the gram stain. When you do this in the lab, this is the step you're going to have to be most careful with. And this is called the decolorization step. And what you'll do is you'll rinse this slide with alcohol. You can also use acetone, but we usually use alcohol. It works perfectly well. And when you do that, the crystal violet is actually very soluble in alcohol. It rinses out of the cells very, very easily. That's why you have to be very careful with the step. You don't want to do it too long. So you rinse with the alcohol wash, and once you've done that, you stain again with a stain, another generalized stain called safranin. And the safranin is, if a cell stains with safranin, it turns bright pink. So we have two colors that we've used. We've used deep purple. It actually turns a blue-black color once you've added the iodine. And then we have safranin color. So the logic is that at this decolorization step, in a gram-negative bacterium, the alcohol actually disrupts the outer membrane of the cell, and it also very easily washes the crystal violet out of the peptidoglycan layer, because this peptidoglycan is so thin. In the gram-positive, the peptidoglycan is extremely thick, it's deeply stained, and the alcohol does not have time to rinse it all out. If you leave the alcohol on too long, however, you can rinse out the crystal violet even from a gram-positive cell. So let's have a look at these slides. These are a mixture of gram-negative and gram-positive cells, and we see the reactions of each. When we do our initial staining, here are gram-negative rods here, and gram-positive cocci. When we do our initial staining, all of them stain. When we add the mordant, all of them remain stained. But when we rinse with the alcohol, the crystal violet rinses out from the gram-negatives only, and it leaves them colorless. Now, if you were to now go and look at this cell, at this slide rather, at this stage, you would find it very difficult to see the gram-negative bacteria because they'd be completely colorless. There's nothing there for you to look at. So that is why we use this. We stain with safranin so that the gram-negative cells become visible as pink cells. These gram-positives also stain with the safranin, but the crystal violet iodine staining is so deeply blue-black that it obscures the safranin. So what you see in a mixed slide like this, gram-positive and gram-negative, you'll see gram-negative pink cells and gram-positive deep blue-black cells. And here's a typical slide stained with the gram stain. And this is, as we saw in the diagram, a mixture. We have gram-positive cocci. They appear as blue-black coloration. And here we can see these rods. These are gram-negative rods. And you will, I'm sure, perform a very similar staining reaction. This is often, as I say, the very first clinical test that one does when trying to identify an unknown bacterium. And first of all tells us the gram reaction, but it's also a very good way of visualizing the form of the cells that you are dealing with. Clearly here we have cocci, and then we have rods that we are dealing with. There are some bacterial groups, however, which present some problems. And this is one of them. This is a genus of bacteria called Mycobacterium, a very, very important genus, especially in human health, but also in agriculture and various other things, because Mycobacterium is a diverse genus, and several of the species cause very serious illness to human beings and to animals as well. And the most important one in terms of human population is Mycobacterium tuberculosis. Here is the name here. This is another name that you're going to have to remember and know how to spell. Okay. Mycobacterium tuberculosis is the causative agent of tuberculosis, and tuberculosis remains one of the most important human diseases worldwide. In fact, after an initial period in the 20th century when the numbers began to fall, numbers of the incidence of tuberculosis fell in the human population, it is now on the increase again and has been for nearly 30, 40 years. So it remains an extremely important disease. The other famous disease caused by mycobacterium is caused by the bacterium Mycobacterium leprae, and that causes the disease of leprosy. There are various other mycobacteria, however, which are completely harmless and are commensal part of our microbiome. The thing about mycobacterium is that the cell wall doesn't only consist of peptidoglycan. They are fundamentally gram-positives, but they do not respond to the gram stain because of the presence of this wax, which is present in the cell wall. And it's a wax called mycolic acid. And it prevents the stain from actually reaching or being able to bind to peptidoglycan or penetrate into the interior, into the cytoplasm, etc. So they just don't stain properly with a gram stain. And instead, you use a special technique called the acid fast technique. And what you do with them is you make a smear and you then heat up the smear together with stain. And what happens then is that the wax actually will melt and the stain is able to penetrate, and you then decolorize with an acid alcohol. And the acid alcohol will rinse off stain from the outside, but is now unable to penetrate to the interior because the wax resets. And what you end up with, you will discuss this in more detail in your lab, but what you end up with are cells which are stained a bright pink color. Any cell that is bright pink like this is immediately diagnostic of mycobacterium, of an acid fast cell. And this is actually a sputum sample here. So this is all debris and mucus and stuff from the sputum, but embedded in it, there are numerous of these long filamentous rods which have stained a distinct pink, and that is diagnostic of mycobacterium. Okay, so let's return to our discussion just briefly about the functions of the cell membrane. And this should be somewhat familiar to you from biology, because I'm sure that you discussed in biology the action of the cell membrane in regulating the transport of molecules in both directions, into the cytoplasm and then from the cytoplasm out as well. And remembering our basic cell membrane structure is a phospholipid bilayer forming a plasma membrane. And when we look just at that, we realize that in fact, some molecules are able to move across this barrier. In particular, things like gases and some ions, but ions actually find it rather difficult to go across, but some may. Water molecules may just be able to make their way across, unaided by any protein. But the direction of travel of molecules across this cell membrane is by purely diffusive properties. So they move from an area of high concentration to an area of low concentration on the inside. by diffusion, by simple diffusion. But there are very few molecules which are actually able to make their way through both a hydrophilic layer of the heads and this dense hydrophobic layer on the interior here. Instead, in order to get molecules to and from across this barrier of the cell membrane, there has to be a system of proteins embedded in the cell membrane, which will actually allow transport to take place. Now, many of these transport systems still depend upon the diffusive properties of these substances. So we're going to talk first about a set of proteins which allow for facilitated diffusion. In other words, they still use diffusive energy to allow molecules to move, but they make it much easier for that to take place. And these may vary a lot in their properties. Some of them are absolutely non-specific, and they will allow the passage of many different kinds of molecules. They just act as a simple open channel. The channel, however, can be controlled, and it is sometimes controlled simply by its presence. If the cell needs lots of these channels, it will simply make them and embed them in the membrane. If it does not need them, it will degrade them so that they are no longer there. But once they are there, they serve as an open channel to allow molecules to move across. And this is in particularly important for ions, which find it very difficult to move across the barriers, or else large molecules, which simply are unable to make their way through. So this is the process of facilitated diffusion. Now, some of these facilitated diffusive transporters are in fact very, very specific. They are specific for particular molecules. And a really typical one is the glucose transporter. Again, it is still using diffusive pressure. It still has to move glucose from an area of high concentration through to an area of low concentration. But what happens is the protein has an area here, which is an active site, which binds only that particular molecule. And when it binds it, the whole protein changes shape. So here you can see there's a cleft into which the molecule of glucose is going to bind. Once it's bound there, the whole molecule changes shape, and the molecule loses its ability to bind the glucose, and the glucose is spat out on the other side. The only energy used is the potential energy, which results from this differential of concentration. There's a high concentration out here, low concentration here. The molecule naturally tends to move in that direction, but this transporter, the facilitator, actually ensures that only the glucose is moving across. It's not just opening a channel and allowing all sorts of things to come across as well as the glucose. It's only allowing glucose to specifically go across. So there is another set of these transporters, these passive transporters, which are also using facilitated diffusive method, but they are specific for water. Now, water can make its way across the membrane, surprisingly enough. It can make its way, but it does so very slowly. And often the cell will require water rapidly and in high concentrations. And so there are a set of proteins called the aquaporins. And the aquaporins are transport proteins, again using facilitated diffusive pressures in order to move water molecules from the outside to the inside. And these are extremely important in water regulation. And if you think back to your biology, you may remember that in eukaryotes, and invertebrates in particular, the aquaporins are very important in the kidney. They are one of the ways in which we regulate the concentration of our blood serums and also our blood pressure by allowing water to move into blood vessels. Okay, so the point about the diffusive and facilitated diffusion, it uses no extra energy. Okay, it only uses the potential energy of that differential high concentration to low concentration. In many cases, cells will want to move molecules in the opposite direction, to move molecules from an area of low concentration to an area of high concentration. This is extremely important because it means that by using methods like this, the cell can begin to accumulate a high concentration of useful molecules inside the cytoplasm and keep them there. So it can work the other way too. Sometimes, as we'll see when we look at how membranes function in establishing gradients of their own, they may want to pump things out as well against the gradient. But in order to move from an area of low concentration to an area of high concentration, you have to use energy. You have to use biological energy. And biologically useful energy is usually in the form of ATP, adenosine triphosphate. And we use the energy contained especially in adenosine triphosphate, has 3 phosphates, and it is that third last phosphate bond which is particularly important. And usually ATP is converted to ADP, adenosine diphosphate. The energy from that last bond is used to do work. And in the case of active transport, that work is for the protein concerned to move a molecule from an area of low concentration to an area of high concentration. This molecule here is a very famous one. It's called the sodium phosphate pump. And what it does is it works both directions. So here, for example, this is sodium, and this is the interior of the cell, and the pump is actually moving these sodium molecules here to the interior of the cell, and it is moving phosphate molecules out. So here's the high concentration of phosphates being moved from the cytoplasm out, and here is the low concentration of sodiums being moved into the high concentration on the interior. So in order to do that, this protein, each time it moves these molecules, it has to use ATP. It has to put in energy in order to do it. So this is active transport. Okay, so I'm going to spend a little bit of time now repeating some of the information about features of the bacterial cell. In some cases, I'm going to do this quite briefly because we did discuss this to some extent in class. So this is going to be something of repetition. First of all, let's talk about the cytoplasmic elements, those things which are included in the cytoplasm of a bacterial cell. Remember this. This is a prominent feature in the cytoplasm, and it is the large circular chromosome. It is wound up and in particularly dense area, which we refer to as the nucleoid, not the nucleus. There's no interior membranes, but the nucleoid. The whole of the cytoplasm appears granular because it's densely packed with ribosomes. There may also be little circles of DNA in addition to the chromosome called the plasmids. And plasmids are very useful because they often carry useful information conferring on the bacterial cell the ability to resist antibiotics, to produce toxins, various other things as well. And when we talk about disease process, we will hear about how some pathological features can be acquired by bacteria by acquiring plasmids. Plasmids can be acquired from the environment if they are in solution, but they can also be transferred by various means. So in addition, in the cytoplasm, we may have all sorts of inclusions. Very often these are things like phosphate reserves, glycogen, which is a fuel. It's a polysaccharide. We also have glycogen, but it's a common storage product in bacteria. Lipids, sulfur granules, many other things may be accumulated and visible inside the cytoplasm. Here is one inclusion which is highly specialized, and this we did not discuss. I think I mentioned it in our first week, but it bears further discussion because these are the endospores. So this word spore, a spore always implies some sort of reproductive cell which is long-lived and resistant to environmental pressures. So very often spores, like for instance fungal spores, are resistant to drying out, to desiccation. They are small, very small, and are easily blown around. And those which land in a suitable environment will germinate and produce they will produce either the fungus if they came from a fungus, or bacterial cells if they're coming from a bacterial spore. However, the bacterial spore is produced under very particular circumstances, and also it has some really strange properties, and which make life very difficult for human beings at times. What I mean by that is, these are resting cells. They're produced especially when nutrients become depleted, and when a bacterial population begins to come under stress. They undergo a particular kind of cell division. Normally, cells divide by binary fission. You end up with two identical cells of just about the same size with exactly the same function. But in the production of bacterial spores, the genome divides as normal, but one part of the genome moves to a particular area of the cell. It varies from genus to genus where this happens. In this instance, it's moved to the end, and a division begins to produce a very small cell. So this is the protospore, if you like, here, and this here is the mother cell here. So we've got a large mother cell which is producing a very small cell at the end, and after division, there's cell membrane, but then a highly specialized cell wall is constructed. That cell wall has numerous layers, and the layers are highly complex. The sets of polysaccharides, proteins, and various other things, which confer very particular properties on the spore. Eventually, the cell will die. The mother cell dies, but the spore is now in a resting state, and is sometimes referred to as the resting spore. And the spore can be released into the environment when the mother cells disintegrate. Here is one here. The interior here is packed. It has some molecules which are specially designed to protect all the bacterial proteins that have been accumulated there. And this is one of the things which confers very long life on these spores. The dense cell wall around them confers on them the ability to resist drying out, for example. Heat, they can be boiled and suffer no ill effects. They are very resistant to bleaches, naturally occurring chemicals like antibiotics or lysozyme or anything like that. They're even highly resistant to radiation. And this means that there's very little that's going to kill bacterial spores. Now, when I tell you that these spores are produced by these two genera, Bacillus and Clostridium, both of these groups have many pathogenic members who produce these spores, which are then liberated into the environment. And this is why they become so clinically important. Because once you have, for example, a population of these bacteria, say in a healthcare setting, they can release spores, which, because the spores are so small, can easily become airborne, also transported by vectors like hospital workers, etc. And these spores, once they settle in a suitable environment, they'll begin to grow. And as they are pathogenic, that suitable environment is very often a patient. They may settle into a wound, they may be inhaled, they may be swallowed, whatever. But when they end up in a suitable environment, the spores will germinate and establish a new bacterial population. And if that is a pathogenic population, then these infections can spread very easily in a healthcare setting by the spores themselves. The process of spore formation is called sporulation. And the spore is an endospore because it is produced internally to the cell. Spores like fungal spores are produced externally to the organism that produces them. But the endospores are produced internally to the cell that produces them. So spores are very important in hospital hygiene and in transmission of disease as well. Okay, some other features I did mention. The exterior to the cell wall, there is often a glycocalyx. And the glycocalyx is composed largely of mucoid proteins and polypeptides, and they take various forms. For example, sometimes they are very well organized and quite tight, close to the cell, and this we refer to as a capsule. Now, the glycocalyx has many different functions. The first important function is it does provide some protection to the cells on the interior. It does shield the cells, for example, from attack by immune molecules, and in fact, from recognition by the cells of the immune system in vertebrates. Here is the capsule here. This is a special staining technique. It's called a negative stain, and what's happened is they've stained the background, and then they've stained the cells in the middle. So these are the bacterial cells, and the capsule is visualized here as this clear area. And the presence or absence of a capsule is often diagnostic for identification of the bacterium, but may also tell us whether or not the organism is pathogenic or not. And you're going to hear of one particular group of bacteria, the Streptococcus pneumoniae. There are two forms. Obviously, Streptococcus pneumoniae, it causes pneumonias. But one strain of the bacterium does not have a capsule like this and is unable to establish infection. The other strain has a capsule and is able to establish infection. And the reason is a second function for the capsule is often attachment, to attach the bacterium to a host cell and begin the pathogenic process. So here's as I said, this is would be considered part of the glycocalyx. The capsule is part of the glycocalyx. Here's a loose glycocalyx around this cell. Sometimes they are so loose that they're easily shed by the bacterial cell and continually produced as a slime. And this is also called a slime layer here in a transmission electron micrograph. These filaments out here are glycocalyx filaments forming a slime layer. Here in this one, they're more tightly organized to form a distinct capsule. I also did mention the flagellum, that this is again a diagnostic feature for bacteria when we're trying to identify them. And there are, remind you that we saw a cell with a bunch of flagella at one end. And there are many different ways in which cells can be flagellated, and that is genetically determined. Sometimes they can, in fact, change the number of flagella that they have. Here is a very common situation, and here is the bacterial cell with flagella emerging from all different parts of the cell. And this we refer to as peritrichous. And a good example would be E. coli. It has peritrichous flagellation. Some, Vibrio, for example, are monotrichous. They have a single flagellum at one end. This, we'd say, is polar. So this is polar and monotrichous. Lophotrichous means a bunch. So here is a lophotrichous flagellation, several flagella at one end. Again, this would be polar and lophotrichous. And this is called amphitrichous. It occurs at both ends. That's where the amphi part comes from. This is spirillum. And spirillum is a bit of a special case. I explain why. For all of these other bacteria, it is extremely difficult to see the flagellum. You can see the cells moving quite easily, but you cannot visualize the flagellum. It is too small. It is too thin. In standard clinical microbiology, if we want to visualize the flagellum, we have to use very, very specialized staining techniques. And I will confess I've done it 1,000 times, and I've probably got it to work about 3 times. Okay, it's just a very difficult staining technique to make these flagella visible for the ordinary light microscope. Very easily visualized, however, on transmission electron microscopy, which is what all of these 3 are. And that's what we would normally do, is use transmission electron microscopy or scanning electron microscopy to visualize these flagella. In the case of spirillum, it's a little bit different, because what looks like a single flagellum here is actually a bunch of flagella all tied together. And they still use the same way for motility, but in spirillum, the flagella are very easily visualized, because this is actually quite thick. Okay, so I'm not going to go into a huge amount of detail about how they work, but just to show you, I told you originally that first of all, the flagellum is itself quite rigid, and it forms a corkscrew shape which spins in space. And this is how it is spun. It actually has a little motor. Here it is here, embedded into the cell wall and into the cell membrane. And it is driven by proton power. Never mind exactly how, but this membrane establishes a proton gradient, as we discussed before, and then uses the potential energy of that proton gradient to create kinetic energy to spin this little motor, so that the whole flagellum rotates like that. But there's only one useful direction for the flagellum to go. So the beating of the spinning of the flagella in the whole cell is coordinated. And when they're all coordinated and spinning in the correct direction, the cell can swim, and it can swim straight. But when they move for a particular reason, right, to get away from something, bad conditions, or towards good conditions, maybe towards nutrients, towards oxygen if they need it, whatever. If they're moving straight towards what they want to go to, and they swim past it, then the flagella begin beating in the opposite direction, and all coordination is lost. The flagella lose their ability to drive the cell through water, and the cell just begins to tumble until it receives a positive signal again. And when it receives a positive signal again, the flagella coordinate again, and it begins moving forward. And so it moves in a series of staggered steps, closer and closer and closer to the conditions that it wants to be in. That closer and closer and closer to the conditions that are giving it the positive stimulus. A highly specialized situation is in the spirochetes. And I remember that I told you that spirochetes are very common environmentally, very common components of microbiota. And this is a free-living one called Leptospira. And the spirochetes, the cell is a tightly wound corkscrew, and it is wound around a flagellum, which runs through the center here. And that is called the axial filament. And here it is, it also spirals around. It functions like the other flagella. It's driven by a motor, but now, because they're wound around one another like this, they actually, when the flagellum spins, it actually causes the whole cell to spin like a corkscrew. And it can go the other direction as well. So these are one of the few bacteria which are able to move forward and backwards. The other bacteria, they only move forward and tumble. Spirochetes are able to go forward and back like this. And it's because of this peculiar relationship of the cell itself to its flagellum. Okay, some other things I did mention, but the first is these proteinaceous components called the fimbriae. These are extensions from the cell membrane, and they usually function to attach cells to the substrate. Another feature which makes them important in pathogenesis, and also in establishment of biofilms, of layers of bacteria. The pili are very similar to the fimbriae, but they are much longer, and the pili are involved in two strange kinds of bacterial motion. Apart from swimming, some bacteria can glide along a surface. They have to be in contact with the surface, but they can glide. But there is another type of motion where the cells actually twitch, like jumping beans, to move. And these are the responsibility of the pili. And to distinguish them, the fimbriae are shorter, pili are longer. There is one very important type of pilus, which is a hollow tube called the sex pilus, which enables the exchange of DNA between two different bacterial cells. We will hear mention of that when we talk about bacterial genetics. Here is the sex pilus here. Now what happens is we're not pumping DNA all the way through here like this. The pilus is used to establish contact with a recipient cell. There is a donor cell and a recipient cell. The pilus establishes contact, and then it shortens to bring those two cells close to one another, and then the DNA exchange takes place. Okay, eukaryote cells, I'm not going to discuss the structure of eukaryote cells. You did this extensively in biology, and I want you to please revise that. By the way, here's another, oh, sorry, although you're going to do your microscope chapter on your own, this is an example of confocal microscopy, producing these very, very beautiful, very detailed images of cellular structure by staining various components of the cell and then visualizing at numerous levels through the specimen and having a computer reconstruct almost a three-dimensional view of what was seen. So everything comes into very, very high focus and with a tremendous amount of detail. The cell, in this case, the cell is actually here. The cell membrane here is lying here. And what you're visualizing here, first of all, this is a eukaryote cell. Here's the nucleus here. And these are various skeletal, proteinaceous skeletal elements of the cell extending out into the body of the cell. Please make careful note of the difference between the animal and plant cells. Those are the two major types of eukaryotic cell. Make careful note of what is present in both, what is the similarity of the two, and what are the differences between the two. Make sure you have that clear in your mind. All of this material that I say to you, you need to revise, you need to do this on your own. This is all examinable, but this is stuff which you should have covered in biology. And as I told you right at the beginning, you don't forget and just scrub out the classes that you have already done, because we don't have time to go over all this stuff again. You need to revise this, but it's there in your brains somewhere. You just need to polish it all up, make sure that you understand clearly. Okay, so there are tests next week, and those are your first tests, and they will consist of about 20 multiple choice questions, 20 marks, and then 10 marks for short written answers. And I'm sure that you're all going to do really well. Okay, and I'll see you next week.